Okay, so Devyn, Tevin, and I all did these chapters together. And I started out with chapter 16, which is on gene expression. So, overview. Given that all cells contain the same DNA, how do cells and organs differ from one another? So the answer to this question, lies in each cell's individual need for unique sets of proteins that allow the cell to properly perform its designated function. Due to the varying protein requirement among cells, only a small number of protein sets are expressed in a cell, as not each cell expresses the same gene transcribed proteins. Expression, occurs when DNA is transcribed into RNA and RNA is translated into proteins. However, not all encoded DNA become RNA or protein due to specialized cells carrying out specified bodily functions. An example would be the heart, lung, and eye proteins. Okay, regulation of gene expression. In order to function properly, each cell must regulate the synthesis of proteins from information encoded in their DNA. This is called gene expression in the two different types of cells. So first we have prokaryotic cells, which lack a nucleus, and transcription and translation occur almost at the same time. In prokaryotic cells, protein synthesis is controlled by DNA transcription regulation, and genes are free floating in the cytoplasm since there is no nucleus. So in eukaryotic cells, DNA is in the nucleus and is transcribed into RNA. RNA is then translated into the cytoplasm into protein. And this can occur at the epigenetic level, transcriptional level, post-transcriptional level, translational level, or post-translational level. The last level involves regulation of gene expression after the protein has been made. Okay, so this image is just a kind of a simple image of a prokaryotic and eukaryotic cell. And as you can see, it kind of goes into the differences between the two, the main one being that eukaryotic cells contain a nucleus and prokaryotic cells do not. All right, so controlling gene expression. In prokaryotic cells, a gene will be synthesized by transcription and translation if needed by the cell. However, in eukaryotic cells, the number of proteins moves up or down at any of the multiple levels where regulation can take place and is also based on the specific needs of the cell. In short, for both prokaryotic and eukaryotic cells, protein levels are altered by the cell's need for proteins. The less a cell requires, the less will be present, and vice versa for larger necessities. All right, so evolution of gene expression. Prokaryotic cells their gene expression is regulated at the transcription level only, and they must complete every cellular task as a unit. However, in eukaryotic cells, gene expression is regulated at multiple levels, and the absence of a cell wall allows these cells to organize together to differentiate into complex organs and tissues. And these adaptations, in eukaryotic cells mean more control, which can be defensive in nature, providing some protection against parasitic or viral invaders. Eukaryotic cells are also more efficient in functioning. All right, so prokaryotic gene regulation. There's 3 types of molecules. So the first one is the repressors molecule. I guess I could have just said repressor molecule, but anyway. Repressors bind to the operator regions of the DNA sites adjacent to the genes they control. And these repressors prevent transcription of a gene because of an outside stimulus. So the second type of molecule is the activator molecule. And activators bind to the promoter sites of the DNA adjacent to the genes that they control, which means that they increase the transcription of a gene because of the outside stimulus. And then the third one is called the inducer, which is produced in the cell or in the environment, and they can repress or activate transcription based on the cell needs, which is kind of what this figure over here on the right shows. It just shows that an inducer switches on and what it does for the cell. Okay, so the operons. Proteins needed for a particular function form operons, and there are 3 types. The TRP operon is responsible and negatively regulated by presence of tryptophan, and pardon my mispronunciation. And these are repressible, which means that they are on by default, but can be turned off by an inducer. Okay, so the second one is a catabolite activator protein, also known as CAP, which are transcriptional activators that turn on genes. And the last one is the lac operon, which is inducible so that it can activate or repress depending on the needs of the cell. Its purpose is to use the lactose, which, lack in the environment, and is inducible, which means that they can be turned on by the presence of an inducer. Okay, so the lac operon, for this to be activated, the glucose level must be very low or absent and lactose must be present. Then transcription can occur. With no glucose and no lactose, no transcription takes place, which this figure on the right is kind of explaining that in, I guess, a more, I don't even know how to say that, but like a more defined way, I suppose. But you can see, it's kind of cut off up here, but it says glucose absent, lactose absent, which illustrates the lac operon. Okay, so what happens with excess lactose? If there is a great amount of lactose in the environment and no glucose, strong transcription occurs in the lac operon. This occurs because the repressor is released from the inducer, a lactose is present. CAMP is high because glucose is missing, and this high level allows the RNA to bond strongly to the promoter, potentiating robust transcription processes, which again, this figure to the right is also illustrating that. But the only difference is up here, it says glucose absent, but lactose is present, which illustrates exactly what I just said in picture form. All right, so this is just another one of the figures that signals that induce or repress transcription of the lac operon. And it just kind of explains what induces or represses the lac operon transcription. Okay, eukaryotic epigenetic gene regulation. Epigenetic variations are variations in gene expression brought on by inheritable or environmental factors rather than modifications in the DNA sequence. Gene expression in eukaryotes begins with gaining access control of the DNA, which is a form of regulation known as epigenetic regulation that occurs prior to transcription initiation. DNA methylation, is one type and histone modification another. Basically, modifications help to regulate whether genes are turned on or remain off. Okay, so this is figure 16.7, derived from the textbook, and it kind of explains what methylation of DNA is and how it affects the nucleosomes. And nucleosomes, as seen in the figure, slide along DNA, and when compactly spaced, which is the top portion of the figure, gene expression is switched off because transcription factors cannot bind the DNA together. When nucleosomes are independently spaced, bottom of the figure, the DNA is uncovered and allows transcription factors to bind, which causes gene expression to occur. Alterations to the DNA and histones influence nucleosome arrangement. All right, epigenetic changes are not always beneficial. They can result in chronic diseases like autoimmune disorders and diabetes, as well as mental disorders and differing types of cancers. Figure 16.6, as shown below, illustrates how DNA folds around histone proteins to create nucleosome complexes, which control protein access to the DNA, whereas part B of the figure uses an electron microscope to show that the nucleosomes upon formation look like beads on a string, which personally, I think that's really cool, because for me, I wouldn't have thought that nucleosomes would look like beads on a string, but as you can see, they do, and I find that really fascinating. All right, so histone proteins mobility is dependent on signals located on both the DNA and the proteins themselves. These signals act as tags to the histone proteins and DNA that instruct the histones as to whether a chromosomal region should be reopened or remain closed. These tags are chemical alterations, acetyl, methyl, or phosphate groups, attached to distinct amino acids to the nucleotides of the DNA or within the histone proteins. The tags do not modify the DNA sequence, but how securely wound the DNA is around the histone proteins. DNA itself is negatively charged, altering the charge of the histone proteins, which unaltered by acetyl groups are positively charged, therefore, alters how securely bound together the DNA molecule is. Although atypical histone alterations are not guaranteed to cause cancer, drugs that modify histone alterations can assist in the treatment of cancer by switching on genes that aid in directing cell division and growth. RNA, also known as ribonucleic acid, interference is vital within cells, which acts as a middleman, quote unquote, that allows genes to code for proteins. However, small forms of RNA can constrain gene expression by linking to separate pieces of RNA or even impacting DNA itself or histones. In addition to histone modifying drugs, RNA interfering drugs are in development that could affect atypical genes in cancer cells. DNA can also be altered, but this happens within precise areas known as CpG islands. CpG islands are expansions with a high abundance of guanine and cytosine dinucleotide DNA pairs, CG, located in the supporter areas of genes. When existent, they cause the cytosine portion of the pair to be methylated, an alteration that modifies how the DNA communicates with the protein, including the histone proteins that manage access to the region. Tremendously hypermethylated, are DNA regions with deacylated, for the pronunciation, histones are securely entwined and transcriptionally stagnant. In figure 16.8, this figure is just kind of showing the epigenetic changes to the chromatin and what might result from that. And it also says what might result in epigenetic changes in terms of cancers and things like that. All right, eukaryotic transcription gene regulation, the promoter and transcription machinery. Transcription in eukaryotic cells first requires RNA polymerase to bind to a DNA sequence for the process to begin. General basal factors are also required to assist in binding the RNA polymerase to the DNA by binding to the promoter region. The promoter region is directly above the coding sequence, which can either be a couple nucleotides or a few hundred nucleotides long. The longer the supporter, the more accessible the binding points for proteins, which allows for better control over the transcription process. Specific transcription factors bind to areas outside the promoter region and work to turn on or turn off the polymerase activity. The TATA box, and I'm not entirely sure that I'm saying that right, but the TATA box is an example of a binding site. What if there's a mutation at the promoter region? Mutations are accidental changes that occur in the DNA, causing problems with transcription factors at the promoter. Can lead to missing or deformed proteins and either can result in disease or be silent, which means no disease. Mutations can be caused by environmental factors such as UV exposure, chemicals, and viruses. This table down here at the bottom left-hand corner is just kind of going more into detail with the examples of mutations in transcriptional regulatory elements. Two types of mutations. There are two types of mutations, and one is the dimerizing, which causes changes in DNA structure that prevents the base pairing needed for DNA replication, while the other one is called oxidative, which causes the formation of free radicals that interferes with the DNA bases, preventing them from pairing correctly. And this figure at the bottom just shows a regular DNA and then incoming UV photons and how that can alter that, or I guess mutate, that would be a better word, but that kind of goes into detail more with a picture. All right, enhancers, repressors, and transcription. Enhancers assist in transcription for some eukaryotic cells. They are DNA sequences that together are called distal control elements. When binding occurs with an enhancer sequence, the DNA must bend to allow the enhancer and promoter regions to come together. When this occurs, increased transcription of a distant gene results. Repressors block transcription from occurring by binding to either promoters or enhancers. They respond to external stimuli to make necessary changes. And like I said earlier, the figure off to the left is kind of going more into detail with that, with an illustration. Eukaryotic post-translational control. Processing, the transcribed RNA means changing the RNA into its mature form for translation, which means that RNA splicing is required in the first stage involving exons, which are where the RNA is coded, and also before leaving the nucleus, introns are removed through the splicing process, and the illustration beneath that point goes more into detail with how the spliced RNA are formed. Okay, and then that goes into achieving RNA stability, which caps keep both ends protected from enzyme attacks. Duration for the RNA to remain in the cytoplasm can be controlled. And this rate of decay is called RNA stability. And more decay equals less translation, and less decay equals more translation. Post-translational modifications affect stability, amount of degradation, and function of the protein. And this figure goes more into detail with the 5 modes of alternative RNA splicing. And if you didn't know, alternative RNA splicing is a technique that permits separate protein products to be manufactured from a single gene when different blends of introns and exons are eliminated from the transcript. So that, like I said, that just goes into the 5 modes of RNA splicing. RNA stability and RBPs. RNA binding proteins, also known as RBPs, can bind to the mRNA above or below the coding region. Above or before is the 5' UTR, also known as the untranslated region. Below or after is the 3' UTR. Which RBP binds determines whether there will be an increase or decrease in RNA stability. If too much of an alternative protein binds the 3' UTR of an RNA, degradation occurs, leading to less of the protein encoded in or to be available for translation. And this leads to mutations. And the image on the right, like I said, for the other ones, goes more into detail with an illustration. RNA stability in microRNAs. MicroRNAs, or miRNA, can also affect RNA stability. They are matured by proteins called dicers. Like RBPs and transcription factors, they bind to a specific part of the RNA, but also can bind with an RNA-induced silencing complex, also known as the RISC. Then the mRNA is translated or degrades depending on cell needs. MicroRNA, this image just kind of goes more into detail with that. RNA binding proteins that regulate pre-miRNAs and, well, I guess I would say miRNAs, I'm not sure how to pronounce that, and pre-miRNAs biogenesis by recognizing sequences or structures on the hairpin RNA. RBPs labeled with green are promoting miRNA processing, and RBPs, or PBPs, colored with orange are inhibitors of the miRNA pathway, while RBPs that regulate the stability and turnover of mature miRNAs. Eukaryotic translational and post-translational gene expression, the initiation complex and translation rate. Akin to transcription, translation is directed by proteins that initiate and bind the process. In translation, the structure that assembles to begin the process is known as the initiation complex. The initial protein to bind to the RNA to launch translation is the eukaryotic initiation factor 2, also known as EIF2, which is active when it links to the hypermolecule guanosine diphosphate, GDP. When bound together, the EIF2 GDP binds the compact 40S ribosomal subunit, where the methionine initiator tRNA connects with the EIF2 40S ribosome structure, guiding with it the mRNA to be translated. And for a continuation of the initiation complex and translation rate, when the initiator complex is put together, the GTP is transformed into GDP and energy is dispersed. Next, the EIF2 protein and the phosphate are freed from the structure, and the massive 60S ribosomal subunit connects to translate the RNA. The connecting of IF2 to the RNA is directed by phosphorylation. If phosphorylated, the EIF2 experiences a conformational adjustment and will not connect to the GTP. When this occurs, the initiation structure cannot assemble accurately and translation is hindered. When consistently unphosphorylated, EIF2 connects the RNA and diligently translates the protein. And just like the other figures, this one goes more into detail with EIF2 and phosphorylation. And just a little summary would be the expression of genes can be directed by factors that connect the translation initiation structure, chemical modifications, protein activity, and longevity. Proteins can be synthetically altered with the insertion of groups including acetyl, methyl, ubiquitin, and phosphate. The insertion or deletion of these groups directs their task or the amount of time they reside within the cell. These alterations can direct where a protein is located within a cell, for example, in the cytoplasm nucleus or attached to the plasma membrane. Synthetic alterations happen in response to external stimuli, including UV light exposure, abnormal amounts of stress, heat, or lack of nutrients. These alterations can modify translation, transcription, epigenetic accessibility, or mRNA stability, which all affects the expression of differing genes. This is an essential mechanism for the cell to quickly modify the levels of specific proteins in response to the environment. Since proteins are included in every aspect of gene management, the phosphorylation, which, if you didn't know, is the addition of a phosphate to an organic compound of a protein, can 1. Modify accessibility to the chromosome, 2. Can modify translation by modifying transcription factor connecting or function, 3. Can alter nuclear shuttling by impacting alterations to the nuclear pore structure, 4. Can modify RNA stability by connecting or not connecting to the RNA to control its stability, 5. Can alter translation, escalation, or de-escalation of, or 6. Can modify post-transitional alterations, which is an increase or decrease phosphates or other synthetic alterations within it. The inclusion of ubiquitin group to a protein targets that protein for degradation. Ubiquitin behaves like a signal designating that protein lifespan has run its course. These proteins are then transported to the proteasome, an organelle designed to eradicate proteins to be degraded. One method of directing gene expression, therefore, is to modify the lifespan of a protein. And this image just goes a little bit more into detail with the ubiquitin and how it designates proteins for degradation. And proteins designated by ubiquitin are transported to the proteasome for degradation. Okay, cancer and gene regulation. When genes are silenced or activated incorrectly by a mutation, normal controls are bypassed and an increased growth rate occurs. Increased phosphorylation of cyclin B proteins is one such post-translational event that contributes to this increase in binding of a transcription factor to its site in the promoter, and it works to cause cell overgrowth. These cancer-causing genes are called oncogenes, which is another topic that we have discussed in earlier chapters in class. Okay, so the altered processes in cancers. Different cancers and different people can have any of these altered processes, and rather than go back into detail too much, I actually just went ahead and listed the sections in chapter 16 that each came from, and I just assumed that if there's any interest, people could go back and look at that themselves rather than me go back again and repeat myself. Epigenetic changes, protein modification, and phosphorylation are examples of alterations observable in cancer cells. All right, P53, the tumor suppressor gene. While there are many tumor suppressor genes in cells, P53 is the most studied due to its mutations in over 50% of all types of cancers. The P53 protein operates as a transcription factor that can connect to the sites in the promoters of genes to launch transcription. The mutation of P53 in cancer, therefore, will substantially modify the transcriptional activity of its selected genes. Proto-oncogenes, such as the P53, are positive cell cycle directors, and when mutated, can become oncogenes and can trigger the production and growth of cancerous cells. So this is an image that I took from Encyclopedia Britannica, Inc., and it just kind of goes into detail about P53 and its structure, and it has the human DNA with it and all of that. So it just kind of goes more into detail with the P53 structure. All right. New cancer therapies. Research into new cancer drugs aims to take advantage of knowing the exact gene modification causing abnormal cell activity. By knowing gene expression patterns, personalized recipes for medication can target the abnormal cell activity while sparing the healthy cells. If a cancer is caused by overgrowth, the drug can help turn it off. If a cancer is caused by a gene that is silenced, the drug can help get it turned back on. These occur through processes such as DNA methylation, histone modification, and RNA interference. These take advantage of the reversible nature of these processes. To reverse the epigenetic alteration process in cancer cells, patients taking medications to turn the gene expressions off could stop taking these medications, which would allow the genes to turn back on due to the inhibitor no longer being present. Specific cancer therapies. Two examples of these personalized medications for cancer therapies are: one, in patients with breast cancer that have high EGF protein levels, the medication can block the EGF receptor to decrease tumor cell growth. And two, in patients with breast cancers that are positive for hormone receptors, like estrogen, can take medications that block those receptors. This helps control tumor cell growth and spread. About 2/3 of breast cancers are affected by hormones. All right, and now we go into chapter 17 with Devyn Nimblett, and it is on biotechnology and genomics.
So, please turn to the slide. Okay, so as a brief overview, genomics is the study of a whole genome, while biotechnology are technologies that use biological agents to further our studies or technologies. And these two are very heavily related, as the application of one can help to advance or help understand the other. Next slide, please. Next slide, please. Okay, so before talking about biotechnology or anything like that, the heart of genomics is being able to study them and manipulate them in order to advance our biotechnology or advance our understanding. Now, okay, so for basic genetic manipulation, so like the controlling of genetic material being DNA and RNA, okay, so in order to understand how to manipulate it, we first have to understand nucleic acids as a whole. Nucleic acids are macromolecules that are comprised of organic molecules. They have nitrogen bases made of phosphoric backbones and of sugar, connected by phosphodiester bonds. And the phosphate groups have a net negative charge. DNA, as we all know, is found in the nucleus of a cell. It is made of many nucleotide sequences. Full sets of DNA are called genomes. A full DNA molecule is found in complementary strands held together by hydrogen bonds. So to begin the manipulation, when temperatures are raised above the optimal range of DNA, it'll denature and the two strands will come undone. It's during this period that DNA can be replicated with the help of DNA polymerases. Once the DNA has been once the replication has occurred, the DNA and temperatures fall, DNA will anneal and reconnect. RNA, however, unlike DNA, is not restricted to the nucleus. It can travel throughout the cell, and there are multiple types of RNA. the most common used in the analysis of the most common used in analysis is messenger RNA or mRNA. It is difficult to analyze, however, due to it being less stable than DNA. Next slide, please. Okay, so in order for any manipulation to take place, it first has to be, DNA and RNA first have to be extracted before they can be analyzed or manipulated in any way. And in order to do so, in order to extract DNA or RNA, the cell has to be broken, and most macromolecules. This is done using enzymes. These enzymes, like protease and ribonuclease, breaks down unnecessary or unneeded proteins or polymers that just aren't required for the extraction. The DNA is then precipitated in alcohol and is then at that point visible as sort of like a whitish gelatin mass. And it can be stored for years as long as it's kept under -80°C temperatures. RNA is primarily used to study the expression of gene patterns. And many of these same techniques that are used to extract DNA are used to extract RNA as well. Next slide, please. Okay, so the first biotechnology we'll talk about is gel electrophoresis. And the purpose of gel electrophoresis is to separate molecules. It separates them by size and by their charge. Well, not by their charge, with an electrical charge. My apologies. Now, it doesn't matter if the fragments or molecules are full or fragmented, they can still be run no matter what. They can still be separated. And because nucleic acids are negatively charged, they're negatively charged ions and are neutral at or basic pH. In gel electrophoresis, the nucleic acids are loaded into slots in a container filled with the semi-porous gel matrix near the negative electrode. There will be, in gel electrophoresis, there are two electrodes, the negative and the positive, both at opposite ends. Once the positive electrode and negative electrode are connected to the container and the charge is applied, the nucleic acids are then pulled towards the positive end due to their negative charge. The smaller nucleotides will move faster, and thereby separation occurs. There can be a size comparison made as long as you are using the molecular weight standard samples. The matrix can be observed using various dyes or stained DNA fragments, and they'll appear like bands at certain distances from the top of the mixture. They will essentially appear as long smears. Uncut genomes are usually too large and remain at the top of the mixture, forming one single, large strand. Next slide, please. Okay, so now that the DNA has been separated, now that it's been extracted and separated, before anything else, it needs to be amplified. And this is usually by PCR. DNA, when collected in bulk, is visible to the naked eye, again appearing as a white gelatin. However, most analysis demands looking at a specific gene or location on the gene, and for this, amplification is required. The technique polymerase chain reaction, or PCR, has many purposes in a lab. One specific purpose is used to amplify regions of the DNA so that it can be further studied. Its other uses are for cloning gene fragments, identifying contaminated or foreign DNA, and amplification of sequencing, which we will talk about later. Using a process called reverse transcriptase, DNA fragments can be amplified from RNA. So even if DNA is not initially extracted and then run, if you have the RNA, using the reverse transcriptase, RNA can essentially become DNA or be amplified to make DNA. By adding DNA nucleotides to the mRNA and the use of the reverse transcriptase enzyme, it can recreate the original DNA strand. Next slide, please. Okay, so hybridization and blotting. Blotting is the process of moving those molecules from the gel in the gel electrophoresis to a nylon membrane. Cellular blotting is specifically when DNA is moved from the gel to the nylon membrane. Please move to the next slide. Okay, so molecular cloning and recombinant DNA. Cloning is essentially just creating a replica, just a perfect replica. Molecular cloning is the reproduction of the cloning or reproduction of the desired region of a genome. The cloning of fragments. The cloning of fragments is what allows genetic manipulation of specific regions and introducing them into bacterial hosts, which we will talk a little bit more about later. Okay, so plasmid. A plasmid, also called a vector, is a small circular molecule that is used to insert genes into via multiple cloning sites, so that that gene can be replicated and used in other experiments. Once a plasmid has the desired gene in it, that plasmid is then introduced to a bacterial host. Bacterial hosts are used because a lot of bacterial hosts or bacteria in general can infiltrate cells of plants and animals and many organisms in order to and replicate their DNA, and hence and as long as they're filled with that desired gene, they can apply that desired gene into whatever the bacterial host is then infects. Okay. The bacterial fragments called foreign DNA or transgene. The bacterium is, like I just said, it has its own host DNA in which the desired gene is then injected into. Plasmids. Plasmids are natural in bacterial population and can offer antibiotic resistances. Researchers have repurposed plasmids for cloning and production of important genes such as human insulin. The multiple cloning sites can be cut with different commonly available restriction endonucleases. Cloning of cancer cells to study them, like an example would be cloning cancer cells. Researchers can clone cancer cells to get a better understanding and manipulate the genes to get an understanding of what gene is causing the cancerous cell, or is it a specific gene, or something pretty much along those lines. Next slide, please. Okay, so restriction endonucleases cut DNA sequences into a predictable manner. They stagger cuts in the two strands of DNA so that they'll have cut ends that have either 2 or 4 base single-stranded. This creates an overhang. Overhangs are capable of annealing with complementary overhangs. These are also called sticky ends. Once added, the enzyme DNA ligase permanently joins the two fragments with phosphodiester bonds. And now any DNA fragment generated by restriction endonucleases can be spliced between the two ends of a plasmid that have been cut with the same restriction endonuclease. Recombinant DNA. Recombinant DNA is a plasmid that has had foreign DNA introduced into it. It is artificially created and does not appear naturally. These are also known as chimeric molecules. This means that the origin points of the molecules originated from different organisms. Sometimes plasmids may need to be moved to another host so that they can better express specific genes. Any proteins that are expressed are called recombinant proteins. Some can be engineered, and then some proteins will be expressed under only specific conditions. Next slide, please. Okay, cellular and reproductive cloning. Okay, so Unicellular organisms clone themselves all the time through binary fission. This occurs due to replication of DNA via mitosis. Okay, now for reproductive cloning, this kind of cloning is the recreation of a genetically identical whole multicellular organism. It differs from regular sexual reproduction, as these cells in sexual reproduction are both haploid and then combine with the different genetic information of both parent organisms. Under natural circumstances, it is impossible for multicellular organisms to produce a genetically identical copy of an organism. Like I just said, this is because through sexual reproduction, the hybridization of these genetics from the two parent organisms are not a direct copy of either or. It is a combination of both. However, with reproductive cloning, it's an asexual reproduction, which leads to parthenogenesis, or virgin birth. This is when the embryo grows and matures without fertilization. As an example, this occurs in species that lay eggs that hatch even if the leg lays unfertilized. This is done by replacing the haplonucleus with a diploid one from a cell of any organism of the same species than the zygote. Of the same species, then the zygote will be genetically identical to whatever donor it acquired. Next slide, please. Okay, so the success rate of reproductive cloning is incredibly low. The first animal to ever be cloned was a sheep named Dolly. Since then, it has worked on horses, bulls, goats, and other organisms. However, there are problems that arise. Often enough, the clones show some sort of abnormality, whether it be facial, limb, or even cardiac abnormalities. Next up is therapeutic cloning. Therapeutic cloning has the purpose of therapeutic cloning is to produce human embryos so that they can be harvested for stem cells. These stem cells could be used to remedy highly detrimental diseases or defects. However, there are a lot of ethical concerns with therapeutic cloning. Next slide, please. Okay, so genetic engineering. Genetic engineering is the alteration of genotypes with the use of recombinant DNA in order to modify an organism's DNA to express whatever genes that are most desirable. Any organism that receives recombinant DNA or foreign DNA is called a genetically modified organism, or GMO. If the DNA comes from another species than that of the recipient, it is called transgenic. Bacteria, plants, and animals have all been modified in this way. GMOs have been in academic settings since as early as the 1970s. Primarily in the agricultural, medical, and industrial fields. In the use of GMOs are commonly a part of the use of GMOs are used in a lot of pre-processed foods. Before modern technologies, researchers determined the function of genes based on the phenotype and then used that to determine what the genotype was, or the genetic basis. Modern technologies, however, allow the researchers to start at the DNA sequence or DNA level. The method called reverse genetics was once used to determine the function of a body part by damaging it. An example is if a fly has its wings damaged, it can no longer fly. So researchers were able to understand that body part has this function based on what the fly could or could not do after its cells and body were damaged. The use of this modern technology has resulted in a process called gene targeting. Gene targeting is the use of recombinant DNA, or again, foreign DNA, vectors, again, plasmids, to alter the expression of specific genes by either adding mutations or deleting a full part of the gene sequence. The same process is now just at the gene sequence level. Next slide, please. Okay, biotechnology in medicine, in the medical and agricultural fields. Medicine. Okay, so in the medical field, we now have a process called genetic diagnosis, which is testing for genetic defects and before treatment is offered. Now this is good because treatment plans can be based off the findings and testing, and genetic testing is offered to fetuses to find any presence or absence of disease-causing genes with specific debilitating diseases. After that is gene therapy, genetic engineering technique used to attempt to cure diseases. Gene therapy is the introduction of a desired gene into the genome to attempt to cure an inheritable disease, introduced via vector in a virus to infect the host cell. Okay, moving on to the agricultural field. There are some recombinant proteins that need animal hosts to produce desired genes. By doing so, the desired genes are incubated. The animals are essentially used to incubate the desired genes. The animals that have been modified are called transgenic animals. They are used to produce the desired genes for testing and mutations. The same is true for plants. Transgenic plants, let's see, they're called transgenic plants. Creating traits like disease, herbicide, or pesticide resistance is the primary use for transgenic plants. They can also be used to improve the nutritional values and improve the shelf life. And this practice has been long running. Next slide, please. Okay. As a continuation of the medical field, most traditional vaccines use a weakened or inactive version of microorganisms to start the immune memorization and response. With modern techniques, however, genes instead of the full microorganism can be cloned into vectors and reproduce bulk amounts of the desired genes. So instead of having to use that full inactive or weakened version of whatever disease it is, we can now code for specific genes to aid in building that resistance. This is used to stimulate primary immune response and trigger immune memory. Moving on to antibiotics. Antibiotics are a biotechnology in and of themselves, with the express purpose to attain advantage or fight bacterial populations. They are produced by microorganisms like fungi, and they can be produced on a large scale by manipulating those fungal cells. Okay, recombinant DNA technology has been used to mass produce human insulin from E. coli. Before this point, pig insulin was used. However, humans had allergic reactions to it. But now that it's being produced in E. coli, now that human insulin is being produced in E. coli, we don't have to worry about that allergic reaction anymore. Now, a continuation of the agricultural side. Transgenic plants are unnatural. So because of this, genetically modified organisms, whether they be animals or plants, are very closely monitored by the government to ensure that they are safe for both humans and the environment itself. Extensive testing is done to ensure ecological stability, to make sure that we don't throw things out of order. Genes naturally do transfer between species in microbial populations. Bacterium like Agrobacterium tumefacans, please excuse my pronunciation, can introduce their DNA into a new host. Agrobacterium, this agrobacterium causes tumors to form in plants. It doesn't kill the plant, but it stuns it. Now, inside of this agrobacterium, it has a set of plasmids called TI plasmids, and they're what the bacterium used to incorporate their DNA into the infected plant. Researchers, by manipulating the TI plasmids, have been able to incorporate desired genes into animals and plants. Next slide, please. Okay, so Bt toxin and the flavor tomato. Okay, so Bt toxin. The bacterium Bacillus thuringiensis, again, I apologize for my pronunciation. This toxin produces protein crystals during sporulation. These crystals are toxic to insects, and when ingested, these insects will stop feeding on plants in only a few hours. And the result is the death of the insect a couple days later. The gene is an incredibly effective organic insecticide. So modern technology has made it possible for the recreation of Bt toxin by other plants. And so it's been cloned and it's been introduced into other plants. In addition, it is safe for the environment and non-toxic to human and or other mammals. So it has been approved for use. Now, a good example of genetically modified organisms in terms of transgenic plants is the flavor saver tomato. The flavor saver tomato was the first genetically modified crop to be added to the market. It was made using RNA technology so that it slowed down the rotting process. As a result of this genetic manipulation, it also had increased shelf life and improved flavor. Next slide, please. Okay, so we've talked about manipulating genes, but now we have to talk about the location of these. Okay, like I said earlier, genomics is the study of the whole genome, and genetic mapping is the process of finding locations of genes on each chromosome. Okay, so a genetic marker is a gene or sequence on a chromosome that co-segregates or shows linkage with a specific trait. So by studying genetic maps, starting with linkage analysis, linkage analysis is a procedure that analyzes recombination frequency between genes and determines if they are linked or show independent assortment. In the past, geneticists have had to rely primarily on the phenotype to determine what the genotype might be. Shortly after this, however, Gregor Mendel brought the concept of genes and observed some traits were inherited together, and speculated that genes were physically linked on the same chromosome. The mapping of genes led to the first genetic map. Okay. Observations come from the offspring. The exchanging of homologous pairs of chromosomes is referred to as genetic recombination. It occurs during the crossing over process of DNA of homologous strands of DNA like non-sister chromatids. Linkage analysis, however, is based on natural recombination frequency. So the further the two genes are, the two genes are from each other, the more recombination is likely. If the recombination frequency is less than 50%, the genes are said to be linked. Next slide, please. Okay, so this slide has a lot of information on it, and I apologize for that. But maps need these genetic markers. Early versions of maps used known genes as markers. Now, markers are more sophisticated and are used to compare the genomes of individuals in a population. Individuals in the same species are genetically similar, but not identical. There are unique sets of traits. The minor differences are good for genetic mapping, however. A good marker is a chromosomal region that shows polymorphism or multiple forms in a population. Some markers are, some specific markers are restriction, fragment, like length, polymorphisms. Again, I'm sorry about my pronunciation. Variable number of tandem repeats and microsatellite polymorphisms, and the single-stranded nucleotide polymorphisms. When the DNA of an individual is cut by endonuclease, RELP, or restriction fragment length polymorphisms, are detected and recognize a specific sequence. Fragments are made, gel electrophoresis is performed. The DNA of every individual has a unique pattern of bands, one cut with a specific set of endonucleocases. It is referred to as the individual's DNA fingerprint. These specific areas of the chromosome that are polymorphic leads to the unique banding pattern. VNTRs are repeated steps of nucleotides in the non-coding regions of DNA. The non-coding DNA has no function, but is still transcribed, and it is considered active. Microsatellite polymorphisms are like VNTRs, but the unit is small. SNPs are variations in a singular nucleotide. Okay. The recombination process is natural. Is natural mapping. It is affected by an increase or decrease in the level of natural recombination. Some areas are hotspots for recombination, meaning that it occurs more. It's more likely to occur there. And due to this, mapping information must be obtained from multiple methods. Next slide, please. Okay, next up is, we've talked about genetic mapping. Now we need to talk about physical mapping. Physical mapping is showing the actual distance between markers and nucleotides. The three methods of making physical maps are cytogenic mapping, radiation mapping, and sequence mapping. Cytogenic mapping uses microscopic analysis of the stained areas of a chromosome. However, it only gets approximations. The distance is not exact. Radiation mapping. Using radiation to break down DNA fragments, the radiation level can be adjustable for fragment size. It is not affected by the increase or decrease of recombination. Sequence mapping. DNA sequencing technology allows for detailed maps, so the distance is measured in the number of base pairs, and it is more accurate. Now, what do we do with all this information? Well, we store them in genetic libraries, or genomic libraries, my apologies, and complementary DNA libraries. These speed up the actual physical mapping process. Using sequencing technology and genetic site making maps. A sequence tagged site, or STS, is a unique genome sequence with an exact known chromosomal location. Common STSs are expressed sequence tags, or ESTs, and single sequence length polymorphisms, or SSLP. ESTs are short STS and are identified with complementary or cDNA libraries, while SSLPs are obtained from known genetic markers and linked genetic and physical maps. Next slide, please. Integrating genetic mapping. Okay, so genetic maps essentially gives you rough outline, and physical maps give more detail, but both share the purpose of being used to study the entire genome. Mapping uses model organisms. Model organisms will be explained a little bit later, but they are used for comparison. Mapping is still a relatively new process, and advancements are still being made even now. The information obtained from mapping is entered into databases to make information widely accessible to both researchers and the public. One database being GenBank. GenBank at the National Center of Biotechnology and Information, NCBI. Next slide, please. Okay, so so we go from genome mapping to now sequencing these genes in genomes. Okay, so strategies for using sequencing in projects. Now, modern sequencing projects use chain termination method, also called the deoxy method. It was developed by Fred Sanger in the 1970s. This method uses a primer and a regular deoxynucleotide, or DNTP, a monomer of DNA, and a monomer of DNA to replicate a single-stranded template of DNA. The primer and the DNTP are labeled using fluorescent colored deoxyribonucleotides. Not deoxyribo, my bad, deoxynucleotides, or DDNTPS. DDNTPs, my apologies. They are missing hydroxyl groups, however. Each one is labeled with a different color. Every time one is added to the grown complementary strand, it terminates DNA replication there. This results in the formation of multiple short replicated strands that have been terminated at different points on the strand. The fragments are then processed using gel electrophoresis. The strands form a ladder due to the different sizes and show the size of the strand and the DDNTPs. Sorry. Due to the labeling earlier, DDNTPs can be identified, and this results in the sequence of template strands. Next slide, please. Okay, so early strategies: shotgun sequencing and pairwise end sequencing. Okay, so shotgun sequencing is sequencing that uses DNA fragments cut into smaller pieces randomly. All are sequenced using the chain method described earlier. Fragments are then analyzed to find wherever overlaps may occur. The whole sequence can be reformed by finding the overlaps. So by cutting these fragments into smaller pieces and finding where the overlap lies, the whole sequence, the whole template can be then reconstructed. Okay, so a contig. Contig is the assembled larger sequence from the overlap. Looking at the overlap information in the fragments, the whole DNA sequence can be reconstructed, as I just said. At first, shotgun sequencing was only done at one end, meaning that only one end of the sequence of the fragments were analyzed. My apologies. This worked for short fragments, but for larger genomes, both ends of fragments are analyzed. This is called pairwise ending. Pairwise end sequencing. It's more complicated, but the reconstruction is easier due to having more information to work with. Next slide, please. Next generation sequencing. Next generation sequencing is the automated sequencing techniques used to sequence DNA quickly. These processes usually use sequencers, and these sequencers are low cost and can sequence millions of short fragments of DNA. In one day, from 25 to 500 base pairs can be sequenced. Although these sequencers are low cost, the software they use, however, is very sophisticated. This is to make sure that they can handle the ordering of the fragments. Next slide, please. Okay, uses of whole genome sequences of model organisms. The first organism to have its genome sequenced was the bacterium, and I forget my pronunciation, Haemophilus influenza, by Craig Venter in the 1980s. Shotgun sequencing was used by Fred Sanger to completely sequence to completely sequence a bacterial virus. It was the virus was FX174 with 5,368 base pairs. This was the first to be completely sequenced. Although the first organism to be sequenced at all was bacterium, this FX174 was the first to be fully sequenced or completely sequenced. Other organelles and viral genomes were later sequenced. For example, the genome of yeast, and forgive my pronunciation, Saccharomyces cerevisiae, due to its large genome size, it had 74 labs collaborate on sequencing it. This started in 1989 and ended in 1996. By 1997, however, the sequenced genomes of 2 model organisms were available. Model organisms are a species that is studied as a model to understand the biological process in other species. The model organisms were a form of E. coli, or K12, and yeast, the same yeast as earlier. Other model organisms such as the mouse, the fruit fly, the nematode, and even elegans, and even humans are now known. This information can be applied to genetically similar organisms, and knowing the entire genome of the model organisms aids in research. Okay, so this moves us on to genome annotation, which is the attaching of biological information to the gene sequence. Annotation aids in experiments in molecular biology by being able to design PCR primers and specific RNA targets. Next slide, please. Use of genome sequences. DNA microarrays are methods used to detect gene expression by analyzing an array of DNA fragments fixed to either glass or a silicon chip to identify active genes and sequences. Almost 1 million genetic abnormalities can be discovered using microarrays. While whole genome sequencing provides information about all the base pairs in the human genome, the study of medical applications of genome sequencing whole genome sequencing can provide information about all the base pairs in the human genome, and this helps in the study of medical applications of genome sequencing, which focuses on abnormal gene function. And knowing the genome makes discovering possible onset diseases and other disorders early more likely. Like we can, essentially knowing more about the genome makes looking out for those types of disorders or diseases earlier on a more likely scenario. This information can be used to help form lifestyle decisions and changes and make specific medicine and medical decisions. Genomics is still relatively new, but in the future, it may become a commonplace to use sequencing to screen newborns for any sort of abnormalities. Genomics can also contribute to the creation of novel enzymes that convert biomass into biofuel. This results in higher crop production and the lowering of the cost of crops. This knowledge allows for methods for better control of microbes to produce biofuels, and genomics can improve impacts on pollutants and pollutants have on the ecosystems and cleaning the environment as well. Genomics has allowed the development of chemicals and pharmaceuticals that benefit both agriculture and the medical science fields. So much effort has been poured into genome mapping and applications similar to it, because many of the benefits and uses for it are extremely useful. The largest issue facing genome sequencing and mapping is the ethics, health, and privacy concerns. If the wrong people get a hold of this information, it could cause a lot of issues. These could potentially, and that is a serious violation of a person's privacy. Next slide, please. Okay, so predicting disease risk at the individual level. Using genome analysis at the individual level, it can be used to predict the risk percentage of diseases in healthy individuals. This information can be used to, again, make changes in lifestyles before the appearance of the disease. It works best when the problem resides in a single gene defect. However, this kind of gene defect only accounts for 5% of diseases in developed countries. This isn't even considering less developed countries. Most common diseases are polygenic or multifactored. It is a phenotypic trait that involves 2 or more genes, as well as environmental factors such as a diet. In April 2010, scientists at Stanford University published the genome analysis of a healthy individual. This individual being Stephen Quake, a scientist also at Stanford University. And it predicted how likely he was to have various diseases. He had what's called a risk assessment performed to analyze the specific percentage of his percentage of being at risk for different medical conditions. And it found a mutation that put him at a risk for sudden heart attack. He was at a 23% risk of developing prostate cancer and a 1.4% risk of developing Alzheimer's. Scientists use databases and publications to analyze genomic DNA so that we can find stuff like this. And if you had a chance to get your genome sequenced, you could have abnormalities discovered and discover possible risk of onset diseases. Next slide, please. Okay, so pharmacogenomics and toxicogenomics. Pharmacogenomics and toxicogenomics, which they're pretty much the same. They're pretty much the same terminology. They evaluate the safety and the effectivity of drugs based on information from an individual's genomic sequence. Responses to drugs can be studied using experimental or live cells before working on humans. And changes in the gene expression provides information about the individual's transcription profile when the drug is present. This can be used. It can also be used as a potential indicator of how toxic an effect drugs might have on an individual. And as an example, genes for cellular growth also control cellular death or apoptosis. When disturbed, they can lead to the growth of cancerous cells. So by understanding an individual's transcription profile or how toxic a drug could be to them, we can avoid instances like disturbing cellular growth genes or cellular growth genes. Genome studies could also help find new genes that are related to toxicity. The personal genome information could be used to prescribe specific medications that'll be the most effective and least toxic to a person based on their genotype. Personalized, which leads into personalized medicine. Personalized medicine is medicine that is specific to an individual based on what will be most effective for them and least toxic based on their genotype. These structures of these may not necessarily be 100% accurate, but they can be tested. Before any symptoms arise. My apologies. Next slide, please. Okay, so microbial genomics or metagenomics. Metagenomics is the study of the collective genomes of multiple species that grow and interact in an environmental niche. These are used to identify new species quicker and to analyze the effect of pollutants on the environment. Metagenomics can also be used to find cures for diseases. Microbiology it is usually taught that microorganisms are best examined under what's called pure culture. Pure culture are conditions that isolate a single type of cell and culturing in the lab. However, pure culturing is not necessarily the best way to culture microorganisms because it can because their gene expression can adapt to the laboratory environment very quickly. Some samples also resist being cultured in isolation because they usually live in microbial communities or biofilms. Next slide, please. Mitochondrial genomics. The mitochondria is an organelle that contains their own DNA. It mutates at a rapid rate, and because of this, it is used to study evolutionary relationships. The mitochondrial DNA and mitochondria as a whole is passed down specifically from the mother in most species, and it is used to trace genealogy. Any information obtained or found at a crime scene has been used as evidence in court cases. Genetic markers have been used in forensic analysis. The first use of genomics in forensics was in 2001. It was a collaborative effort between institutions and the FBI, like genetic institutions and the FBI, to solve the anthrax case specifically. And thanks to microbial genomics, they determined that it was a specific strain of anthrax, and then from there, learned how to counteract it. Next slide, please. Okay, so genomics in agriculture. Using genomics in scientific research, it can reduce trials and failures. To an extent, it can improve crop yield and the quality of crops themselves. Linkage traits or linking traits to the gene specifically, flash gene signatures, which help improve crop breeding and generating hybrids with desirable qualities. Using genomic data to identify desired traits, those desired traits can then be transferred. Genomics can improve the agricultural production in both quantity and quality. As an example, producing crops with drought resistance. Next slide, please. Okay, so basic techniques in protein analysis. This is called proteomics, or I'm sorry for my pronunciation. The study of proteomes or proteomes, the entire set of, the study of proteomes. And proteomes is the entire set of proteins produced by a cell type. The goal of proteomics is to identify or compare the proteins expressed from a genome under specific conditions, and then to study the interactions between other proteins. The information is used to predict cell behavior or make specific drug targets. Much like the technique of genome sequencing, proteomics requires techniques to analyze proteins. The technique for protein analysis is called mass spectrometry. Again, I'm sorry for my pronunciation. It is used to identify and determine the characteristics of a molecule. It has allowed researchers to analyze and determine the characteristics of a molecule. Using X-ray crystallography, this allows researchers to determine the actual three-dimensional structure of the protein crystal at an atomic resolution. One other imaging technique, called nuclear magnetic resonance, or MNR, uses the magnetic properties of atoms to determine the three-dimensional structure in an aqueous solution. Microarrays are also used to study interactions between proteins, as stated earlier. Next slide, please. Okay, so large-scale adaptations of the 2-hybrid screen has made the foundation for the basis of protein microarrays. Okay, so computer software is used to go through the vast amounts of data generated from these analyses. And let me see. Systems of biology. The study of the whole biological systems on interactions within systems. Both genomic and proteomic scale analysis are part of systems biology. Institutions like the European Bioinformatics Institute and the Human Proteome Organization, HUPO, are establishing tools to sort through these vast amount of data on systems of biology. Proteins that are a direct product of genes and demonstrate at the genomic level. Proteins are a direct product. Sorry, my apologies. Proteins are a direct product of genes and demonstrate at the genomic level. So naturally, proteomes are used to compare the protein profiles of different cells to identify proteins and genes related to disease processes. Most pharmaceutical drug trials target proteins specifically for that reason. The information of proteomics is said to identify novel drugs and their mechanisms or actions. Okay. The issue of using these techniques for protein analysis is that it has difficulty detecting small amounts of proteins. There has to be a large amount, or difficulties can arise. However, mass spectrometry is good at detecting small number of proteins, but any variations in expressions can make it hard to discern. Expression of proteins in disease states, my bad. Proteins are naturally unstable. This makes proteomic analysis more difficult than genomic analysis. Next slide, please. Okay, so cancer proteomics. The genome and proteomes of patients that have specific diseases are being studied to gain a basis for that disease. The most prominent one right now is cancer. This is to improve early detection and screening by identifying the protein whose expression is affected by the disease. Let's see, biomarkers. Biomarkers are proteins that are uniquely produced in a diseased state, which and protein signatures are just a set of those proteins in a disease state. For biomarkers or protein signatures to be a good candidate for early screening and detection, they must be excreted in a bodily fluid. So through the blood, sweat, urine, any of those. This is so that large screenings can be done in a non-invasive way, which leads to the issue with biomarkers: false negatives. False negatives are an incorrect test result that should be positive. The issue with biomarkers for early detection is that they have a high false negative rate, causing many cases to go undetected. This makes biomarkers unreliable. An example of used biomarkers is CA-125 for ovarian cancer and PSA for prostate cancer. Protein signatures may be a bit more reliable for detection, and proteomics can be used for individual treatment plans. Predicting if an individual will have a response to specific drugs and possible side effects, proteomics are also used to predict possibility for reoccurring. Next slide, please. Okay, so institutions like the National Cancer Institute have developed programs to improve detection and treatments for cancer, while other organizations like Clinical Proteomic Technologies for Cancer and Early Detection Research Network are efforts to identify protein structures for various types of cancers. And the biochemical proteomics identifies protein signatures and design effective therapies for those cancer patients. Next slide, please. And that was my chapter.
All right, and now we're getting to chapter 21 with Devyn Shelton, which will start a brand new video for him because he had to work today, and that's when we had the time to do it. So we will be starting a brand new video with him in chapter 21 on viruses.